love to welcome Peter Eckmann to the floor, and we can get started with your talk on the automated screening of COVID-19 preprints.
Okay, so hi everyone, I'm Peter Eckmann from UC San Diego, and today I'll present automated screening of COVID-19 preprints. Can we help authors to improve transparency and reproducibility? So for some background, we've seen preprints grow in popularity since COVID-19 first emerged. And as you can see in this graph on the right here, preprints are published at about the same rate as peer-reviewed papers, especially early on when they were actually published at a higher rate than peer-reviewed papers. So we're seeing that speed of publication is really useful during the pandemic, but the lack of peer review of preprints have been concerning for a lot of scientists. So here's an example of a preprint that describes a small number of patients with COVID-19 very early on in the outbreak. The authors decided to withdraw this preprint because they realized that basing any sort of decision on such a small sample of patients is not a good idea. But if the authors never withdrew the paper for whatever reason, how would readers know not to trust it? So in general, we're seeing that quality control is a significant issue with preprints. So as a group, we asked, can we evaluate preprints at scale without relying on authors to withdraw preprints when needed, or the knowledge of readers to discern good or bad science? So to do this, we set a goal to automatically evaluate COVID-19 preprints for reproducibility criteria. And we did this by downloading, parsing, and analyzing preprints using the following tools. So SciScore screens for rigor criteria defined by NIH and the key resources that were used, like cell lines or software tools. ODPub, open data detection and publication, screens for the presence of open data and open code statements. Limitation recognizer screens for self-acknowledged limitation statements. Bazooka screens for bar graphs that were used for continuous data. Jetfighter screens for rainbow color maps, which can be hard to read for colorblind viewers. Seq and Blastin screens for correctly identified nucleotide sequences. And Trial Identifier screens for and verifies clinical trial numbers. So this is a general overview of the pipeline that was used. So you can see up here, we used preprints from bioRxiv and medRxiv, which are the two most popular preprint servers for COVID-19. And we pulled preprint PDFs from these servers. And from these PDFs, we extracted the images and the texts from them. So from the images, we used Jetfighter and Bazooka, the two image analysis tools, figure analysis. And for the text, we used Trial Identifier, Limitation Recognizer, Open Data Detection, and SciScore. And we combined the results from all these tools into a single HTML report, which we then published to Hypothesis, which is a web tool that allows others to see the report in front of the preprint. And we also had a SciScore Reports Twitter account, which we used to share these reports. And we also implemented a study type classifier, which classifies papers as either a modeling paper or a non-modeling paper to change the criteria that SciScore uses to judge papers on. So let's first talk about the preprint data set. So we use bioRxiv and medRxiv, which are, as I said earlier, the two most popular repositories of COVID-19 preprints. And they have a set of preprints hand-curated for COVID-19. And it's updated daily and allows for programmatic access. So on the right here, you can see the web page for that. So now let's talk about the images and text extraction. So here's an example preprint page. And these are the things that we extracted from it. So the red text that's outlined here was removed using a custom algorithm because it's boilerplate text that we don't need, like page numbers or line numbers. The green text was then extracted using machine learning algorithms to pick out the correct sections, and those fed to SciScore, ODPub, Limitation Recognizer, and Trial Identifier. And the images, like in blue, were extracted and analyzed by Bazooka and Jetfighter, the two figure analysis tools. So finally, we can talk about how the reports were published for others to see. So as a group, now that we have these reports, we wondered, how do we let users know that they exist? And how can we let people see them? So first, we thought about using the comment section on bioRxiv and medRxiv, but it turns out that's heavily moderated and they don't allow bots to use it. So instead of that, we decided to make the results available through Twitter and Hypothesis. So we realized that the preprint webpage has a section called tweets referencing this article, which shows all the tweets which link to the article. So we could use that. And we also used Hypothesis, which is a web annotation tool, to essentially display the report that we generated in the preprint side by side so people could compare them. So in the center here, you can just see an example preprint webpage. And down here, you see the tweets referencing this article section. So for this one, our tweet was the top tweet, and viewers can click on this link here to view the report, which is on the right. So you can see the report has, the rigor criteria, the key resources, open data, limitations, trials, and all the other tools are down there. And users can also click on our Twitter account here, where they can see all the reports that we've generated. So we analyzed a few thousand preprints, and this is what we found. So about a quarter of the preprints contained an ethics approval statement, which suggests that the majority of papers are secondary analyses or modeling studies, not primary studies. And only 7% of the papers that use cell lines described how they were authenticated or kept free of contamination. And also only 20% addressed sex as a biological variable, despite there being known differences in how COVID-19 emerges between different sexes. And that's actually one of the reasons we started this study, because the percent was so low. We thought that's a problem. And 6% use model organisms, which were mainly mice. In terms of transparency, about 34% included self-acknowledged study limitations, 14% shared open code, and 14% shared open data. And in terms of data presentation through figures, about 8% used rainbow color maps, which are not colorblind safe, and they can create visual artifacts for viewers with normal color vision. And about another 7% use bar graphs for continuous data, which can lead to misleading figures. And it's better to use something better like a box plot or a histogram to show that. And in terms of what we saw from Twitter, the automated tweets have been viewed about 380,000 times. They currently average about 1,000 views and 10 link clicks, which is people clicking in to see the reports per day. And over time, the account has accumulated 2,500 link clicks, about 100 retweets, and 40 replies. So down here, you can see some of the replies that we've seen, and they range from people telling us that the algorithm is checking for the wrong criteria, as in our tools missed something that was actually included in the paper, like this person. So that would be a false negative for our tools. Some people use our tools for further analysis and improvements of their paper, like over here. And some people raise questions about if automated screening can replace peer review or just work alongside it. So for next steps, we want to run a study to compare COVID-19 preprints, non-COVID preprints, and peer-reviewed publications, and analyze the differences. So you can see like a sample study design here, what it might look like. And you might expect papers that are peer-reviewed to be of higher quality than preprints, but that's what we want to study and see what actually turns out there. And we also want to run trials to determine if giving authors more direct feedback, like through emails or something, can improve reporting rather than just putting it out publicly for everyone to see. Okay, thank you for your attention on behalf of our group. If you want to see more about our tools, you can visit our website here.
Thank you so much for that great presentation, and how timely and how valuable this is for the scientific community as well. We have a couple of different questions, and one of them is, does SciScore also screen for mice and antibodies?
Yeah, it does. It screens for those resources in the key resources table, along with cell lines and those tools.
Okay, fantastic. And the second question that had come in is, will you be registering the study via a registered report?
Yeah, we will be. We will be doing that.
Okay, has it been published already or as a preprint, or are we not at that stage yet?
It's we had a correspondence to Nature Medicine, and that's being published right now.
All right, congratulations.
But we're planning on doing a more in-depth study with comparisons and all that in the future.
Fantastic, thank you. One of the people in the chat mentioned that it seemed like a good use case for PubPeer as well, and I was wondering if you could comment on that.
Yeah, so we've worked with groups kind of like that to put our reports into other websites like Society. I don't know if you ever heard of that. But yeah, we're planning on expanding how our reports show up farther than just, Hypothesis and Twitter, but to let other groups and websites use the reports as well and post them.
Well, thank you. I have one personal question for you. I've also tried to use Hypothesis for a study before, and I found it a little problematic. And I was wondering if you could share any of your experiences using that particular tool.
Yeah, so what we use for it, we use the API, so we don't actually, manually put the reports in. It's pretty easy as long as you don't want to, edit it or anything. Viewers can just click on the link and it just shows up on the sidebar right there. They don't have to do anything. They don't have to know how the tool works. But yeah, maybe using the tool can be a bit difficult, like to edit stuff and that kind of thing.
Okay, well, thank you so much for your time. I got a question that I will answer in the chat myself, but we really appreciate this, and thank you so much for your patience as we were working things out. For those of you who are in this room, we're going to briefly adjourn out of the presentation mode, and then we are going to come back in about a minute to start on our lightning talks. So we'll see you soon. We do also have lightning talks in building 2, and we will try to be starting them simultaneously. So we will see you soon, and thanks again, Peter, for your presentation.